Hello, and thank you to the hosts of the 2022 Hereditary Neuropathy Foundation Thrive Summit. My name is Omid Karkouti. I'm the co-founder and chief operating officer of Rarebase, a public benefit biotechnology company based in Palo Alto, California. And I'm here to talk about or provide a prompt for the question, can existing drugs be used to treat CMT or hereditary neuropathies? Rarebase was founded by patients, patient advocates, biotechnology entrepreneurs, and technology entrepreneurs as a public benefit company to try to accelerate the discovery and development of treatments for rare disorders with a focus on neurological disorders. And our leadership team includes myself as co-founder and chief operating officer, Anna Faber, as CEO, and our Chief Scientific Officer, Dr. Chris Moxham. As I personally know, because of a genetic disorder in my family and all the folks on the line here as either rare disease patients or patient advocates, the genetic disorder is really not, in terms of clinical treatment, a dead end in most cases. There are only a few hundred treatments for rare diseases in total, despite there being 7,000 of them. And as a company, we want a world where a diagnosis can also include a solution that addresses the underlying disease biology. So you can imagine something like, in a future state, a search engine that a doctor could use that could actually tell them about treatments for genetic diseases. And we hope to build tools, technology, and data to support that future goal. And as a company, our mission is to provide a path forward, as in a research path forward, and once we get there, hopefully a treatment path forward for anyone with a genetic disorder. Part of this topic, and to address the prompt, is to identify new uses for existing drugs. And there's a couple of examples here. I won't belabor the point. Maybe some of you have heard about pharmaceutical repurposing before, but the three key examples I wanted to share, there's osteoporosis drug raloxifene that was approved in 1997. It's now used in invasive breast cancers. There's a drug that some of you may have heard of called rapamycin or solimus that was originally approved for organ transplant rejection in 1999 and currently is either indicated or used off-label in a number of rare disorders because it's a downstream effector of a central cell signaling pathway, and so it's used in tuberous sclerosis, ALPS, and other rare disorders, including tumor syndromes and tumor disorders. And then the third is tamoxifen. It's a pretty old drug for breast cancer, a hormone type of therapy, and a new use of it was in the treatment of bipolar mania, was discovered about 30 years later. So it is possible to use existing drugs for new things. And part of what we're engaged with Hereditary Neuropathy Foundation in terms of research is to do that for hereditary neuropathies. And as a company, our mission is to provide a path forward for anyone with a genetic disorder. So within the scope of this process and our goal for looking at hereditary neuropathies and CMTs is to find existing FDA-approved drugs that can be used to treat CMT disorders. The fastest path to applying the results from our research is to identify existing approved drugs that may be in a pharmacy down the road that can be applied. That said, it's possible that for certain genes that cause hereditary neuropathies or CMT, there may not be an approved drug that has a potential off-label use. And in those cases, our company will work towards the invention of new drugs that can be used and investigated for those genes. And during the course of our research, we are looking to ask and answer a couple of key questions. First, what drugs could work for CMT? And second, how can this be tested in cell cultures in a dish to determine whether any of these have their predicted potential benefit? And how we do so is with a drug discovery platform that our company has developed called Function. And Function works by identifying drugs that act on the root cause of genetic disorders by modulating their gene expression, as in the amount of gene or the dose of gene that's present in cells. And that can be directly on the root cause gene that causes the disorder, or on some other genes that are within a network or a pathway that may compensate for the deficit. So there's a cartoon below that talks about or describes DNA and a variant or mutation in the DNA that's represented on the lower track with a pink line. The DNA is transcribed to messenger RNA, which is then translated to protein, and because of the mutation in the lower track, there's a disruption to either the function of the resulting protein or the level or the dose or amount of that protein that's in cells. And in this metaphor, using plant genetics or a flower, in the unaffected or wild type flower, it has a full complement of petals, but in the mutant or variant setting, it has fewer. And so this is a metaphor for the effects of a mutation on gene expression or gene function that's presented with a different phenotype. So describing our drug discovery platform at a research level really consists of three main pillars. First, it's the generation and study of cell-based models of rare diseases, genetic diseases, and that's what we depict on the left are neurons or nerve cells in a dish that are labeled with multiple different types of markers, and then behind that panel there's an animation of the signaling of those cells as represented by the changes in the levels of calcium being measured, and that's a type of assay or test that can be done to look at nerve cell function in a dish. Second is a system of data that's generated from the screening of drug libraries. And with the current scope that we're working together with Research on Hereditary Neuropathy Foundation, Rarebase is currently screening 4,000 drugs on neurons or nerve cells, and generating from each of those experiments approximately 10,000 data points that describe what happens to gene expression within those cells, with a complement of FDA-approved drugs, as well as other experimental conditions that are intended to mimic the effects of interruption or disruption in genes on gene expression, so cellular disease models. And then the third component is the use of computational systems, artificial intelligence and machine learning that can interpret the information from the cell-based models and also the drug screening to predict hits or drugs that may be able to compensate for the disruption of different genes, and also hopefully be able to help predict those that could lead to a favorable response from patients. Now, how the computer modeling gets us hits, there's three aspects to this, a suite of different software tools that we have developed or are developing to interpret the data from our drug screening. The first is called Hit Finder, which ranks the best therapies based on a series of different criteria, some of which are based on the scientific data and some of which are based on the properties of cells, for example, deliverability, side effects, how readily available they are they available in the United States, have they been prescribed to children, in what setting, things like that. And based on the data from the function drug discovery platform, we'll be looking for those hits that can rescue or restore a deficiency that's introduced by a genetic variant in an individual gene. The second is Pathfinder, and in the center of this slide, you see described a cellular pathway where a gene is involved in and acting on a number of different other proteins and producing a protein that's acting on a number of different proteins in the cell, transcription factors and other genes that are essential. And in the mutation, and that known mutation causes a disruption that leads to a deficit in the, in this case, a hippocampal neuron or a brain cell. And Pathfinder is a method where we look for an opportunity to, for an alternative route to lead to either production of that essential protein or to compensate for it through a different manner. And third is model matcher, which is the ability for us to map model cells in the lab to observable or relevant phenotypes or things that can be measured in patients, such as biomarkers, so that we can, from our data, we're predicting drugs that will have a potential therapeutic benefit in treating the root cause of the gene, and also that can be measured when they're being studied in by the doctors and scientists that will be conducting the clinical research portion of this work. So to the second question, how do we test these drugs in models of CMT? First, we're generating nerve cells in our research labs, and we're engaged on doing that for hereditary neuropathy genes at the moment with HNF, and open to collecting specimens for other genotypes. At the moment, we're generating materials for PMP22, both duplication and deletion, MFN2, CNT-NAP1, and C12or65. And how this process works is a blood sample is collected in a nearby lab. Through various steps in cell culture, there are stem cells that are generated through a process called reprogramming, and these stem cells are called iPSCs, induced pluripotent stem cells, from those blood samples. And through research methods, those cells can be differentiated to neurons or nerve cells. And those are the materials that we can use that are disease models derived from patients, where we can test the drugs that we predict from our computational methods and determine whether they have the results that we're hoping to achieve with them. And so beyond the genes that we're currently generating these materials from, we're also banking additional samples for general research. So please do contact your representative HNF for more information about how you may be able to participate in this research. And later in the year, with those cells in hand and with the predictions from our computational models, we'll have results from experimental work of screening of approximately 4,000 drugs, including a full complement of FDA-approved drugs, and then also additional data from disease models from another 1,500 genes involved in neurodevelopment or neurological disorders. And our computational system, HitFinder, will be ranking those best therapies, and those will be tested in the cells, in the patient-derived cell-based models, in a dish, in a paradigm called a clinical trial in a dish, where we would look for testing those compounds in patient-derived cells to see if they have an effect that can ameliorate or reverse the disease state. Our team consists of engineers, scientists, and patients and patient advocates that are working in this field with a hope to identify in the near term potentially effective therapies from new drugs, and in the intermediate to long term develop new medicines where existing medicines do not fit or are not available. And we're honored to work with the Hereditary Neuropathy Foundation and patient communities like yours in this effort. thanks very much for the opportunity to present to you today, and I look forward to further discussions and collaboration with the CMT and hereditary neuropathies patient communities.